fast and loose here and just give you a big picture overview of essentially my entire academic life. But I thought I'd actually start right at the very beginning, because one of the recurring themes of my life and arguably lessons of my life is plan what plan. It's not like I was born thinking I wanted to be an academic. I was born sitting on that, well, not born on that rug, but spent some time sitting on that little rug there. That's my parents, who they were unlucky enough actually to grow up during the war, the Second World War. So they both left school by the time they were about 14, probably essentially as soon as you could, and went off to work and really had no understanding of what academia was. And no one in my extended family has any idea where the brains that me and my sister seem to have came from. Perhaps I absorbed them from that rug I'm sitting on there, because no one really knows. My parents were not lucky enough to have the chance to demonstrate any brains, but there's me at various stages of growing up. And I guess by the time you get to the picture on the right, and I probably 5 or 6 or something there, I'm beginning to realize, yeah, I really enjoy this math stuff and just seem to be quite good at it. And that's really been the one plan I have had, which is I enjoy doing this, so let's do more of it. So I'm going to give you the entire career path, starting at the very beginning, which is as an undergrad, although I won't really talk about this, but I did a mathematics degree at the University of Warwick way back in 1980, early 80s. Warwick at the time was competing with Cambridge to be the strongest math university in the country. And I went there not knowing anyone, had a great time, but at the end of that time left academia essentially and went to earn some money at a place called Standard Telephones and Cables, which was in my hometown, where I was a systems analyst, computer programmer. I lasted for about 6 months before I got incredibly bored. I actually remember the first thing they ever asked me to do there was solve an algorithm, a little bit of code that somebody had written for them that was bugged. And I dug into it for several weeks before I found out what the bug was, just to prove that some of the stuff you learn in statistics is useful. Somebody had implemented Newton-Raphson incorrectly, and it was causing this bug. When you dug down deep enough into this code, you discovered the only error was with the way they'd implemented Newton-Raphson. So if you just did that correctly.
Hold on, Paul. We're having sound
audio equipment.
Sure. Let me know when you're ready.
Now, but we're trying to figure out how to make you louder.
yeah.
Let me see if I can do anything at my end.
It's okay for people in the front, but I don't know. Maybe I'm just getting too old.
Loud as what'd you say?
Yes,
I've turned my input volume up. Does that help at all?
That helps.
that is
All right.
better.
That's better.
Is that enough, or you want more?
It's more.
More? I'm getting close to maximum now, so it may start distorting.
Yeah, do the maximum. Yeah, that's perfect.
We like it.
Club, good.
All right, that's
Okay.
working better. Okay.
Thank you.
All right, so from Standard Telephones and Cables, as I said, I lasted about 6 months, was incredibly bored ultimately, and so decided I wanted to go back to university, but I had a gap, so I ended up driving taxis and working in a farm shop for a little while. That's a picture of me in a farm shop where my mum worked. That's my mum and my sister again, selling overripe bananas. For some reason, we always had loads of overripe bananas that we had to sell, before going back to Cambridge to do what essentially was a master's. Cambridge doesn't call it that for reasons that aren't worth going into, but essentially you're doing a master's. And then from there, ended up doing a PhD in the University of London. Again, deciding academia wasn't for me, so getting a real job as a systems analyst programmer again. This time I lasted, I think, about a year before I got incredibly bored and decided I wanted to go back to academia. And from there, did 3 postdocs and ended up at USC. So I'll tell you various bits of that story, while also picking out some of the bits of mathematics that I have most enjoyed during that time. So I'm trying to extract a few lessons here as I look back on my own academic life. And so this is lesson number 2, how not to choose a PhD mentor. So I'm not going to talk about my life as an undergraduate at all. But after doing my master's at Cambridge, I decided I wanted to do a PhD. And I actually decided I wanted to do it in London before I decided what I wanted to do. And the reason for, and that's a really bad strategy, but I got very lucky and it worked out for me. I'd always lived in and around London, typically 20 miles outside London. And I loved going to see football. That was Chelsea, my favorite team. Going to see bands, going to see movies, and I'd go to London to do that, but I would always have to worry about the last train, which in those days left at about 11 o'clock at night. And you would typically miss it if you went to see a band, which made life very awkward, and you'd end up getting very long bus journeys home and get home at 3 or 4 o'clock in the morning. So that was not fun. So I decided life would be much easier for me if I was a student in London, because then I could just get the last tube home, and that left much later. So I went to London University, University College London in particular, largely for reasons of geography. But I was lucky enough to be interviewed by a guy called Peter Donnelly. He was at the time the most junior member of the faculty, probably only the most junior member of the faculty would have accepted me. But and I was his first PhD student. He went on, he was an absolutely brilliant guy, remains the most, certainly one of the most intelligent people I've ever met. Went on to be the youngest professor in the UK, the head of the statistics department at Cambridge, or was it Oxford? I can't remember which, Oxford, I think. And I essentially lucked out. He was a great person to work for. And I think a lot of it was that we just connected immediately. He was Australian, he loved sport, he loved movies. So we would just talk about those things nonstop. And so sometimes you just luck into things. And so if you do find a mentor that you feel some sort of connection with or excitement to be with, don't be afraid to follow that. So let's talk a little bit about what I did while I was with Peter, because this remains probably the most beautiful bit of math I've done and I've studied in my life. And the reason I've been doing this for 30 years is that I love math. And most importantly, or most particularly, I suppose, I find math beautiful in certain instances. Not always, sometimes it's a pain in the butt, but sometimes it's absolutely beautiful. And this process that I'm about to describe is the one I studied with Peter Donnelly. It was actually even pre-Human Genome Project. So we were beginning to think about collecting genetic variation data, not even sequence data at that point, and then trying to explain how it got there and see what we could infer from that data about human ancestry and the history of the human population. So this is a little schematic to motivate the model I'm about to describe. And so in this figure, can you see my mouse, by the way? I don't know whether you can see it or not.
Yeah.
Okay, great, thanks. So in this figure, each blue oval is an individual, and we'll talk about what that individual might be in a second. And each row is a generation, and the arrows point from parent to offspring. So, for example, you'll notice immediately that individuals only have one parent, which is not how real life works, except in certain instances. And one of those instances is mitochondrial DNA, which you essentially inherit uniquely from your mother and not from your father. So you can think of these blue dots as being females and tracing this picture being a schematic of an evolving population of females where arrows point from mother to daughter. You'll notice there are many simplifying assumptions I'm making here, not least there's only one parent, but I'm also drawing this process as if generations were discrete, as if the population size was constant. So I've got about 25 individuals probably in each row here. Of course, in reality, it's much, much bigger than that. It turns out, for reasons I can't get into today, that these assumptions generally don't matter. The model I'm about to describe is very robust to all of these assumptions. It changes in certain ways for some of the assumptions, but fundamentally it's largely unchanged. And so here's the motivation for this model, which is something we're going to call the coalescent in a second. And it's to observe that what you do in reality when you're trying to study a population is you collect a sample of individuals from that population. So here I've got the present day population in the bottom row of the screen, and I'm imagining that we've sampled the red ones. So here, this is a toy example, so I'm imagining we've just sampled these 6 individuals out of this population size of 25 or so. And then you observe that individuals have parents, and every now and again, 2 individuals have the same parent or the same grandparent or the same great-great-grandparent. So in this particular realization of this process, these 2 individuals here share a mother. And these 3 individuals actually share the same grandmother. So as you go back in time, the number of individuals' lines of ancestry, these little red lines that you're following, never increases and occasionally decreases. So it starts here at 6, but it immediately decreases to 5 lines. Again, because I'm drawing this so that if it's on the page, it immediately decreases to 4 lines and then 3 lines, etc., etc. And then you do a little bit of math and wave your hands, prove some theorems, and show that ultimately there is, with probability 1, there will be a most recent common ancestor. Somewhere in the history of this population, there is an individual from whom all members of your sample have inherited their genetic material, whatever that is. So for our made-up example here, let's imagine it's mitochondrial DNA. These 6 individuals have all inherited their mitochondrial DNA from that individual back there. Furthermore, no one else in that figure has made any contribution to the variation you see in your sample. So if you want to describe or build a model that describes the evolution of the 6 individuals and how they got to be the way they are, you only need to describe this process that was in red on the previous figure. You don't need to worry about all the blue guys. And that's really useful because in the human population, there's a lot of these blue guys, right? Our population size is very, very large. Our sample size for whatever sample you've collected is typically much smaller. It won't be 6, of course, it will be bigger than that, but it's a much smaller object. And so back in 1982, which is about 5 years before I started doing my PhD or something like that, this guy, John Kingman, who was at Bristol, I think at the time, in the UK, derived the theory for this process and called it the coalescent process because these lines coalesce, stick together at certain points in the past and never become unstuck. So when I started my PhD, I remember Peter giving me 3 papers. In those days they were hard copies, of course, just dropping them on my desk and say, go away and read those and come back to me when you're ready and say something intelligent about them. And this was one of those papers. There are actually a couple of papers of Kingman that I had to read and another paper that I can't remember at this point. So Kingman derived the mathematics for this process. And I'm not going to give you the formal definition of it, but I'm going to give you an informal definition, which is that the picture I drew on the previous slide was discrete. In reality, of course, time is continuous. We don't all have offspring at the same time. We have offspring essentially whenever we feel like it, as it were. So in reality, life is continuous. So what you do is you take this discrete process, you think about what happens as the population size goes to infinity, and then Kingman's insight is that if you let the population size go to infinity, you have to measure time in units of 1 over n, where n is the population size. So you let the population size go into infinity, and at the same time, you transform the time axis as well. And if you do it in that way, these pairs of lines of ancestry end up coalescing in continuous time as an exponential process. So any given pair of lines, so any particular pair of red lines here, let's just say this one and this one, will coalesce. The time to coalescence will be an exponential random variable with rate 1 if you scale time in units of 1 over the population size. So you don't need to follow that. You can just take that as given by God. If you want to understand the math, you have to go and read those papers by Kingman or a more recent review paper. Or now there are books. There's a nice book by John Wakely on coalescent, for example. So these lines of ancestry are all coalescing independently to the next event. And so if there are currently k lines in your sample, then there are k choose 2 of these pairs. And so the time to the first coalescence is the minimum of k choose 2 exponential processes, which is another exponential process that has parameter k minus 1 over 2. And that's the coalescent. The time to next coalescence is distributed as an exponential random variable with parameter k choose 2, where k is the current number of lines of ancestry. So 6 at the bottom of that figure, then going down to 5, then going down to 4 until it gets to 1. And when a coalescence event occurs, you pick a pair of liners at random and say that those are the 2 that coalesce. So that's actually all there is to the coalescent. It's a very simple thing to describe in this very most basic form. But as I've mentioned, this most basic form is very robust to many of the simplifying assumptions that are made. The 2 that break it are lack of population structure and only having one parent. And so there are more complicated versions of this process for those contexts. But you can immediately derive some really interesting results. So you can think about what's the expected value of the total height of that tree? How long am I going to have to wait on average to find the most recent common ancestor of my sample? Well, it's just the sum of a bunch of exponential expectations or expectations of exponential random variables. So the math isn't very complicated. It's just that expression there. Where remember, little n is your population size, your sample size, sorry. And if you do the math, you get a lot of nice cross cancellation, and you discover that this expression is just 2 times 1 minus 1 over n, where n is your sample size. So think about what happens to that as n, your sample size, goes to infinity. This expression limits out to 2. So this process, no matter how many individuals you have, has an expected height that's finite. And it happens to be 2, and that's kind of cool because 2s pop up all over the place in the coalescent world. You can also think about the total length of the tree. And if later on you're trying to build a model of, let's say, evolution of DNA, the length of the tree is the total time over which mutations can occur and appear in your final sample. So that's something to do with the expected number of mutations you'll see in your sample. And it turns out that expression is also quite simple. It's just k times the expected value of the expected value of the exponential random variable summed over all the possible values of k. So you get this expression. This one, though, interestingly, and I find this really beautiful and interesting, limits out to 2 times log n. So even though the expected tree height is finite, whatever the sample size is, the expected tree length goes to infinity. And I just find that's a very interesting process to study. You've got something that has finite height, but infinite length on average as the sample size goes to infinity. So that's a cool result. I'm going to give you 2 or 3 other cool results. This next one begins to address why people care about this process. Why do we care about a process that looks at a sample of individuals when what I really want to do is make an inference about the entire population? So let's go back to that motivating problem. If we're looking at the time to most recent common ancestor of a sample or the entire human population, if my data is mitochondrial DNA, this individual was known as mitochondrial Eve, the maternal great-great-great-great-grandmother of all of the mitochondrial DNA in the human population. So there's another result due to Saunders, Tavray, and Watterson. So Tavray is the Simon Tavray that some of you know, who used to be at USC. Both Tavray and Watterson are Australian, and for some reason this entire field is littered with Australians, Peter Donnelly being another one of them, who all were at Monash, funnily enough. This result looks at what's the probability that the most recent common ancestor, so this individual from whom we all descend, of the sample is the same as the individual you care about, the most recent common ancestor of the entire population. So here, this is again a beautifully simple expression. It's not a trivial thing to prove. You have to read this paper to see the proof. Little n, remember, is your sample size, and big N is your effective your population size. And if you're familiar with the term, it's actually the effective population size. Let me start with the right-hand piece of this expression, big N plus 1 divided by little n plus 1. Well, your effective population size is a very large number. So this n plus 1 over n minus 1 is essentially 1. So we can sort of ignore that in terms of trying to get some intuition here. So let's just focus now on the left bit. Well, this is little n minus 1, little n plus 1, where n is the sample size. So let's imagine you have a small sample of 100 individuals. This expression is 99 over 101. And this expression is roughly 1. So even if you only have 100 individuals, the probability that you're the most recent common ancestor of those 100 individuals is the same individual as the one who's the most recent common ancestor of the entire population is essentially 1. It's about 98% or 0.98. Even if you only had 10 individuals, this probably is around 80%. So you need a ridiculously small, shockingly small to me, sample size in order to have a reasonable chance that the most recent common ancestor of that sample is the same as the most recent common ancestor of the population. So that's beautiful result number 2. There are some caveats. This is assuming the population is randomly mating and the human population is not randomly mating. So this term effective population size was used to describe a random, or the size of a randomly mating population that would have equivalent sort of levels of genetic variation to the actual human population. And the number that was typically used was about 10,000. But that's still big enough for the right-hand side of that expression to be 1. So that's another random, beautiful result. I have nothing to do with that result, but it's another result that I think is A, beautiful, and B, tells you why this is a useful process to study. Here's another result. I find this one shocking. To this day, I've been studying the coalescent on and off for 30 years, I suppose, at this point, and I still don't understand this result, how it comes out to work like this. So first of all, let's look at the figure on the right-hand side, which I've just borrowed from a review paper written by Noah Rosenberg and Magnus Nordberg, who both passed through USC a dozen years ago or so. This is just 4 realizations of coalescent processes. So the coalescent is just a Markov process. We don't know what it looked like. So here we're just simulating 4 potential realizations, sampled at random from the family of all possible realizations. And I want to make a statement about these potential different realizations. In reality, the human population had 1 realization. Maybe it looked like the second one, maybe it looked like the 4th one, but you can see they look very different from each other. Because these last few lines coalesce like exponential random variables with low rates, rates 1, 3, and 6 for the last 3 lines, or the last 3 coalescence events, there's a lot of variation in these final few lines. There is much, or in the time taken for these final few coalescences to occur. And you can see here's one in which they happen to take long times, and here's one in which they happen to take much shorter times. There is significantly less variation for the earlier coalescence events because these are exponential random variables happening at much higher rates. So A, this process varies a lot from realization to realization, and that's a sobering thought when you're looking at mitochondrial DNA and you only get to observe it once in the human population. But that's not the thing I want to focus on here. I want to focus on a different result, which looks at how many individuals descend from the last two lines of ancestry that exist before the final coalescence, before the mitochondrial eve or most recent common ancestor was reached. So on this particular realization, I followed the left of the last two branches and I see there are 1, 2, 3, 4 individuals descend from it. I followed the right branch and I see there are 1, 2, 3, 4, 5, 6 individuals. This realization happened to only have one individual descend from the leftmost of the last two branches, and the other 9 all descended from the rightmost, etc., etc. So unfortunately, I've forgotten who proved this result, but I find this result staggering. It turns out that the number of descendants, or the distribution of the number of descendants of the last two lines of descent is just uniform. And to this day, it blows my mind that this comes out to be uniform. And if anyone could ever give me a good intuitive argument for why it's uniform, I would love to hear it. The proof is only about 3 pages long from memory, but I can't remember where it was, so I haven't been able to give you the reference for it. But it's an absolutely beautiful result, and it's results like this that kept me studying this process for 20 and 30 years, because it's a beautiful thing. I'll give you one more beautiful result. Paul Joyce and I, top right there, who's sadly no longer with us, wrote a little review paper in which we talked about a variety of things, but we mostly used it as a chance to just talk about bits of the coalescent that we found intriguing. So here's another result I found intriguing. And I apologize for the fact I'm drawing these trees a little differently on every figure, but they fundamentally, topologically, they're equivalent to each other. So here I'm looking at a tree, and I'm just looking at the length of what we call the external branches. So the external branches are branches that go down to a member of our sample and then go back in time until that line of ancestry coalesce with some other line. Why do we care about those branches? Because mutations that occur on these branches are only represented once in our sample. So this is telling us, if I look at the expected length of these branches, and then multiply that by the mutation rate, that's telling me the expected number of singleton mutations I should see under whatever scenario generated this particular realization of the coalescent. And so here's another example where this magic 2 number turns up. It turns out that the expected length of these external branches is 2. The external branches is 2, regardless of, and this is the key bit, the population size, the sample size. It doesn't matter how many samples there are in this tree. If I drew you a tree with 100 individuals, or 2 individuals, or 5,000 individuals, the expected length of these external branches is 2 in every single case. Another absolutely, to me, mind-blowing result. And that's where math is at its most beautiful, I think, when you get this process that's simple to describe and gives you these beautifully elegant answers for reasons you just can't, at least I can't comprehend in any intuitive sense at all why it should work out that way. You can do the math, you can show it, but it's another thing to actually understand, well, why? Why does it work like that? So that's, regardless of the sample size. So that was me doing coalescent things, and they weren't all from that period in time, but that's just to give you a flavor of what coalescent did. At the end, to return to lesson 1, plan what plan, at the end of doing my PhD, actually postdoc, I went back to do a postdoc with Peter in which we studied this process. A little piece of paper landed on my desk, or I came in one morning and found this piece of paper on my desk, telling me that Bob Griffiths, who was another Australian studying the coalescent, was looking for a postdoc in Melbourne, and did I fancy going down there? So I decided that would be a fun thing to try. So I went down there and studied again the coalescent with Bob Griffiths. We actually there studied the version that's actually appropriate for nuclear DNA when you've got 2 parents and you get 1 copy of your father's chromosomes and 1 copy of your mother's chromosomes. So that process became known as the ancestral recombination graph, and I'm going to give you a reference to a couple of papers I wrote with Bob in a second, but I want to doff my hat to Dick Hudson, because he actually first thought of this process. He did it only for a 2 locus version. Bob and I generalized it, but the initial insight is really due to Dick Hudson, who also wrote the most popular coalescent simulation algorithm called MS of that time. So Bob and I studied this process. That was back in the days where if you're a postdoc in math, 1 paper a year was considered par. We actually only wrote 2 papers over 3 years, but they remain 2 of the best papers I've ever written. Or let's say we've ever written, because we both wrote both of them. Life lesson number 3 turns out to be, so the first paper here, an ancestral recombination graph, was all the math behind the ancestral recombination graph, and is probably the favorite paper that of my own that I've written in my life. We decided to publish it for reasons I don't even remember at this point in a conference proceedings journal, which is a really bad choice, right? Because this paper, it managed to get cited a few hundred times. It's absolutely foundational in this field. You don't put those papers in conference proceedings, but we were mathematicians. We didn't care about that sort of stuff. We went to that meeting. It was a really cool meeting. We presented the results. So we thought, okay, let's publish it there. The other paper, I have a slightly different, interesting story for. Well, actually, let me back up to the first paper. So the first paper was the math. I work on this paper with Bob for probably a year or so, I suppose. And the way it worked is the same way it used to work with me and Peter. I'd study it for a week or two. I'd go and talk to Bob, tell him how far I'd gotten, what I was stuck on. He'd give me some insight. Bob was also an incredibly bright guy that would help me solve my problem. And I'd go away for another couple of weeks and we'd just iterate that process until I had a paper. So I walk into Bob's office one day with a draft of this paper, probably didn't have the same title at the time, and asked Bob to just take it home and have a look at it. So he goes home that weekend on Friday night, reads the paper, decides he really likes it, but feels guilty that he hasn't actually done much of the work. Now, he'd given me all sorts of insights without which I would not have been able to do the work, but the most of the math had been done by me. So he decides, being Bob Griffiths, that he is going to add some stuff to that paper. So over the weekend, he writes an extra 2, 3 or 4 pages at the end of the paper and then gives it to me back to me on Monday and says, yeah, I really like this paper. We should send it off to publish it. By the way, I've added a couple of extra results. And I go away and look at the paper. I take it home and read it and realize I no longer understand my own paper. Bob's results are way beyond anything I can understand. And it takes me, I don't remember how long at this point, but it took me quite a period of time to get back to the point where I understood my own paper. The other paper was much more due to Bob than me. This was coding up an algorithm that let us sample from recombination graphs conditional on observed sets of data. And it was the first one of 2 algorithms of its type, the other one actually being due to Joe Felsenstein, who I've got a little picture of there, who was at Washington at the time. This was a computational algorithm. This became cited even more widely because it was one of the first 2 of its time. The interesting story there, though, is we wrote this paper up, we sent it to a more sensible journal, the Journal of Computational Biology, where people could actually read it. And one of the people who read it was Joe Felsenstein, who had the other competing algorithm, and we were friendly rivals with that group for a number of years afterwards. But he looked at that paper, read it carefully, and then wrote to us and said, by the way, do you know that what you're doing in that paper is you've reinvented importance sampling? What you're actually doing, if you phrase it in this or reframe it in this particular way, can just be viewed as a version of importance sampling, which actually was something I didn't even know of at the time. So it's fun to have someone read your paper and say, well, fun in inverted commas. Yeah, it's a nice paper, but actually this method you're using at least already exists, even though it hadn't been applied in that particular context. All right, so a very similar experience at the end of that postdoc. I come in one day and Bob tells me Simon Tavare at USC is also looking for a postdoc. Would I like to go and do one in Los Angeles? So life lesson number 4 is you shouldn't really do 3 postdocs. It is too many. People start to ask why you don't have a proper job yet. But it's also a hell of a lot of fun. For my money, at least, being a postdoc is one of the best times in your academic life because you just get to focus on the research problems you care about. Although for Simon, I did some teaching as well, which I'll come to in a minute. It is a lot of fun moving to different countries and working. It's also very hard, but you learn a lot about yourself. So typically don't do 3 postdocs, but you can survive doing that many if you're as lucky as I was. So I came over to USC and I continued to work with Simon on things related to the coalescent, and I won't talk about any more of those, but I wanted to earn more money and Simon wanted me to earn more money because I was getting old at this point. I was about 30, I think, by the time I came over here. So he had me teach as well. I can't even remember what the course was, but it was some sort of basic math course. So life lesson number 5, public speaking is hard, right? Doing this stuff, teaching, giving seminars is very hard. And I'm very confident that at that point in my life, I was a terrible lecturer. I know I was always very nervous when I spoke. I know to this day I still speak quite fast, but in those days I would speak way faster than I do now. I must have been hell to listen to for the poor students. So if you're also in that boat and you're worried about the fact that you hate public speaking, you hate teaching, know that most of us feel the same way. But the easiest way to get over that fear is to teach a course. Because if you're getting up 3 times a week, in those days I was just doing 3 one-hour sessions a week to teach a course, you have no choice but to get over that fear. You can't get that nervous 3 times a week. It's too exhausting. So at some point you just come to terms with it and you get better at doing it and become a better public speaker. So the only way through that fear is to just do it a lot, and eventually it gets easier. With Simon, I worked on, and I can see I'm going to run out of time here, so I may not get to the third topic I was going to talk about, but we'll see. With Simon, this now was well into the Human Genome Project era. We were continuing to look at things to do with human variation data, but we also started to look at other applications to triplet repeat diseases, for example, with Norm Arnheim, and to models of tumor growth with Kim Sigmund of our division and Daryl Shibata, a friendly pathologist. We were using a variety of methods, including importance sampling, Markov chain, Monte Carlo, and rejection methods. So for no particular reason, I've picked on rejection methods to use as an example. So if you haven't met rejection methods, they're a very simple algorithm described using these 3 steps. You have some data set D, and you have some model that you think describes how the data got there. For example, the coalescent model of human evolution, that has some parameters, 1 or more parameters, which we'll call theta. And what you're trying to do, being a Bayesian, is construct the posterior distribution of theta given D. So you just iterate this scheme over and over again. You sample a parameter value theta from your prior, pi, for that parameter. You then accept that value of theta with probability D given theta, and return to 1. You just iterate. So when you accept a value of theta, you put it in your virtual pocket. You run this algorithm for as long as you can bear to wait. And then you end up with, let's say, 10,000 values of theta in your pocket. And those are samples from, and they're independent samples from, the posterior distribution of theta given D. So they're your approximation to the posterior distribution that you actually care about. It's a nice algorithm. It has a number of issues that mean it doesn't always work very efficiently, but it, at least in theory, is a very nice algorithm. The problem is that it demands you do the second step, in which you accept this value of theta that you've sampled from the prior with probability P of D given theta. And if your model is complicated, and in the era of big data, your models start to get quite complicated quite quickly, it can be very difficult or even impossible to calculate the probability of D given theta in closed form. So that's the problem we were struggling with at the time, me and us and many others. And so we eventually hit upon a different idea, which is to replace the calculation of D given theta in that algorithm with 2 steps that end up being equivalent. So you start as before, you generate a parameter theta from the prior for theta. And then rather than calculating PD given theta, you simulate a data set, which I'm going to call D prime using theta. And then you compare it to the observed data D. And if the simulated data is identical to the observed data, you accept that value of theta that you used to generate it and you put it in your pocket again. And now you iterate this algorithm over and over again until you've got as many values as you want, let's say 10,000 values of the theta in your virtual pocket. And again, they are independent samples from the posterior distribution. So this algorithm that now crucially involves no calculation, so you can do it in situations in which you don't have closed forms for that likelihood function, still gives you the same result. It's giving you independent samples from the posterior distribution. And this algorithm is actually very old. It's really a version of what was called likelihood simulation back in the day. And you can find that mentioned in a paper by Diggle and Grattan, who some of you may know from probability textbooks, in 1984. So it's an old algorithm. It has a problem though, which is that if your data set is complicated, even if you simulate data under the right model with the right parameter value, you're probably never going to see a data set identical to the observed data set. Let's imagine we ran human evolution thousands of times. You're never going to get exactly the same set of data at the end of that process. So the probability that D prime equals D is essentially 0, even if you're generating under the right parameter value. So this is where Simon and I and others began to essentially invent a field that became known as approximate Bayesian computation, because we realized we could never insist on exactitude here. We had to allow some form of approximation. And there's a variety of ways of doing that. I'm describing one particular way of doing it here, which just makes a small change to the third step. So I still sample my theta value from the prior. I'm calling it theta prime now. I simulate a data set D prime using theta prime. But now I calculate some summary statistic or set of summary statistics that describe key features of the data. So this is where you need some insight as to what those things should be. And that's an entire can of worms that I'm not going to get into. And now, rather than insisting that the data set in the simulated data is identical to the observed data, I'm just going to insist that the summary statistics that I calculate for the simulated data take the same values as the summary statistic for the observed data. And that makes life much easier. Let's just imagine that the summary statistic was just the number of mutations, for example. I have a much better chance of finding a match for that single summary statistic across simulated and observed data. But you're paying a price, which is you're now, and you have to do some math here, but you're now generating samples from the distribution, the posterior distribution of theta, given the summary statistic value and not the data set. So if you live in a kind universe, and we don't, you would be in a situation in which theta was sufficient for, S was sufficient for theta, in which case your posterior for theta given S would be the same thing as the posterior for theta given D. But there are very few real life examples for which that's true. And so in general, you don't have equality here. And one of the big issues with approximate Bayesian computation, which has grown to be very, very popular as a method, is that you cannot, a priori at least, say how close f of theta given S is going to be to f of theta given D. And that's sprung up an entire literature addressing that problem, because it's crucial to have some sort of handle on that, which I can't address in the time available to me now. I'll give you one more example, just because this is my last bit of math that sort of blows my mind to this day. I remember Simon and I were writing MCMC algorithms for recombination processes, and they were absolute beasts to try and run. They would take weeks to run. We'd get results. We never really were confident that we had a bug-free algorithm. And eventually we sort of threw up our hands and said, what we need is some sort of ABC version of this, some sort of approximate version of MCMC, because if you're familiar with the Metropolis-Hastings algorithms, and here's a short specification of one, you end up calculating this thing called Hastings ratio, H, and it involves this same expression, this ratio of it involves some other terms as well, which we don't need to worry about today, but it involves a ratio of these likelihood terms. And so if you can't calculate these things, you can't use Metropolis-Hastings MCMC anymore. So I remember Simon coming back from some meeting somewhere, and he'd been stuck on a plane for hours, and I've never been able to work on planes because there's not enough elbow room, but he somehow manages to do it. And he came back very, very excited because he'd begun to work on the ABC version of this algorithm. And so this eventually got published in this paper, in which I'm first, but I'm just first because this is the mathematics tradition in which you go in alphabetical order. The bulk of the work in this paper, and most definitely the initial insight, was due to Simon. And I want to acknowledge that. This is the MCMC algorithm without likelihoods that he'd begun to figure out on that plane journey, and we refined over the next few weeks or months. It's a very similar algorithm. You sample your theta value. You have this thing called a transition kernel, which says, how do I propose new values of theta given old values of theta? We won't go into what that is. But now, like before, you're using a simulation step in which you generate data using your sampled value of theta. So again, I'm generating a d prime given theta prime. And then you either move or don't move. Well, so let me say it differently. You continue to calculate the Hastings ratio if your simulated data is equal to or identical to your observed data. And if it is, you then go on to calculate this expression. Now, if you compare this to the original version of MCMC, this expression is the same as this bit of the Hastings ratio. Somehow this step 2 and 3, generate data set and then compare D prime to D and see whether they're equal, is replacing this expression here, even though it only seems to involve the top bit. You've got your theta prime, you simulated a data set D prime and said, is it equal to the observed data? So you do lots of math, and we published this paper is in PNAS, I think, which is a really tough journal to publish in, so I recommend not trying to publish in PNAS. So I've told that story already. Well, actually, let me rewrite the algorithm before I finish the story off. So that initial step of simulating a data set and asking whether the simulated data D prime is equal to the observed data D can be replaced or put into the Hastings ratio as an indicator function. So now this algorithm looks more like standard MCMC because I haven't got that sort of rejection step there. It's actually implicit in this calculation because this indicator function will take the value 0 if D is not equal to D prime. So then you can never accept this value of theta prime. This expression here is actually replacing this purple box over here. And this is the bit that I never understood. It took me years to understand how is it that this indicator function that appears only on top is replacing this ratio of likelihoods. Why don't I have another approximation on the bottom of this thing? And it wasn't until somebody called Mark Beaumont, who's top right there, who's actually as much an ecologist and an evolutionary jock as he is a statistician, to my knowledge, although I've never actually met the guy, published a paper that explained to me why this algorithm works. This is another example in which you publish a paper using methods you've been working on for several months, but there's still a piece of it you don't really understand. And it takes someone else to come along and say, oh, you were doing importance sampling. Or Mark Beaumont said, no, actually what you were doing is calculating this expression on the left, which does have, so I'm using L tilde here to just denote some estimator of the likelihood of d prime given theta prime, your proposed value of theta, or d prime given your theta, your currently accepted value of theta. You are actually using two estimators, but there's an important caveat which he was smart enough to work out, which is that you have to reuse here on the bottom, this estimator of the likelihood function, you have to reuse the one that you used, or the value that you used when you accepted theta in the first place. And because I'm going to run out of time, I'll let you think about why that means it disappears in the version of the algorithm that Simon and I derived. I've taught many courses on ABC, or at least the same course many times at this point. This is proof by example of the fact that it's popular. So there's all sorts of issues with ABC. It's sort of built on a house of sand, essentially, in some sense. But nonetheless, it has become a very, very popular method. And so this is a graph in which the y-axis is a log scale, showing that over those first 15 years or so, the growth in ABC papers, largely, well, theoretical and application papers, was essentially exponential. It tailed off a little bit, and I stopped updating this figure when I stopped teaching that course in about 2020, so I can't tell you what it's doing now. But this is just to point out that ABC is a widely used method. On the right-hand side, this is a little dated now, is the cautionary note that being popular is not the same as actually being any good. So you can decide for yourself whether that's a method you'd like to use. But it has been widely embraced by many, many people, and so I'm going to have to skip over some of the applications in which I've used it. In particular, I'm going to have to skip over the tumor growth work that I've done with Kim Sigmund and Daryl Shibata, but hopefully at some point Kim will do one of these and she can describe some of that for us. I'm going to focus on an example from animal behavior just because it was a hell of a lot of fun to do. And one of the life lessons, which is very much a personal life lesson, is that I enjoy academia much more when I'm trying to do a bunch of different things. I get bored very easily, rather than refining the same method over and over and over, even though I would argue that that second method is a more successful way of embarking on or conducting an academic career. But variety for me is the spice of life, so I've always enjoyed doing lots of different things. So I'm going to skip the story of getting a job at NIH by telling a dirty joke, because I'm not willing to repeat the joke in these politically more enlightened times. I ended up going back to USC and deciding to stay at USC. And I just want to point out that I stayed at USC. They both seemed like good jobs to me. Coming to Biostats or going to work at NIH both seemed good, but I was very happy in LA, and I just decided to stay in LA because I was happy there. And I liked being happy, and I didn't know if I'd be happy when I went to DC, which is ironically where I live now, very happily. So again, it's another instance where I sort of followed my gut and got lucky. So I'm going to skip over these for the sake of time, but just to motivate the fruit fly example, this was work due to Sergey Nuzdin and Brad Foley, amongst other people. Sergey runs a lab in CompBio. Well, actually not in CompBio anymore. It's in the other half of that department on the main campus. And he would do do a lab meeting on Friday nights at 4 or 5 o'clock. I can't remember when it was. In which he served alcohol, and it was on my way home. So I would habitually stop by there. I would sneak out from the department at about 3 o'clock in the afternoon, drive over to main campus and go and sit through whatever this talk was and drink wine while I learned something about biology. And one of the stories they told me was this story in which they have cameras pointed at little petri dishes of agar mix on which there are fruit flies, and sometimes off which, because they're tracking these fruit flies as they join and leave these patches. And in the old days, that used to involve a student sitting there taking notes, but at that point in time, which is about 10 years ago now, I suppose, technology was advanced enough that you could film these things. And so these were the sort of films they were collecting. Here's a picture of fruit flies on a patch. They are free to leave the patch as well, so you'll see the numbers change from time to time. And they were just trying to count social interactions. So on the right-hand side, you have what I came to realize much later was very much a cherry-picked example of an algorithm trying to track the location of these fruit flies and then put a little red X on the screen every time they got close together and were having some sort of interaction. And fruit flies have a wide and varied social life, at least in comparison to rats, who, if you put them in the same small environment, will kill each other. But fruit flies will hang out, and if they're male fruit flies and there are females, they'll try and mate with them. Females will try and hang out together. Male fruit flies will fight each other for access to the female. And so they were collecting this kind of data, and they were building models to describe the way these sort of data sets evolved. And I'm not going to get into this at all, but you've got red female flies, blue male flies, the 4 yellow patches, and these flies are leaving patches or joining patches at rates that depend on what they see. on the patch. In particular, if males see females, they'll head straight for them to try and mate with them. If females see males, they'll probably run in the opposite direction because it's not fun having male flies constantly trying to mate with you. And they would build these models and then try and conduct inference of these models, but they were at a complete loss for how to do that because these models were entirely intractable. And so over the course of a few years, largely Brad and I sat down and implemented actually ABC versions of MCMC that allowed Brad to conduct inference for these experiments. And what comes out of an ABC analysis is a posterior distribution for what in this example was a model with 7 free parameters. So Brad had to look at this. This is a 2-dimensional summary of the joint distribution of each pair of parameters and try and work out what is the story that this picture is telling him. And it turns out there was a story that Brad could see about how males and females were interacting as a function of the different lines of flies we were studying. But to this day, I'm impressed that Brad could look at a plot like this as a biologist and come up with a mathematically and biologically coherent summary of what was going on. All right, so as I said, I'm going to have to skip application 2 with apologies to Kim and Daryl. Kim and I, with Daryl, have been working on this on and off ever since I joined this division, and it's been great fun, and I'm really grateful for the work that we've done together, but sadly, I can't cover that, it turns out, in the time available. Instead, I'm just going to go to my closing slides. Well, I should at least mention that I'm part of the IMAGE grant over the last 7-8 years, I suppose, at this point. That's also been great fun, but you'll hear a lot of talks about work from the IMAGE project, so I'm not going to talk about those here, except to mention what's been one of my favorite parts of that project, which is developing this Happy Scientist seminar series, which Kim now runs, in which we teach ourselves things that we feel like we ought to know but don't. And I encourage you to attend those seminars whenever you see them, and I think we have 2 coming up in the next few weeks. I just wanted to close with closing thoughts. So that's a very high altitude view of my life as an academic. What do I feel I've learned? First of all, I want to say thank you to the students, and I see 1 or 2 names on the attendees list that I recognize. It took me a while to realize what a privilege and honor, and most often actually pleasure, it is to be part of educating students in their life in academia and at university. I particularly want to thank the students who sit on the front row or turn their cameras on in Zoom meetings, because I learned as a lecturer that there are very few things more soul destroying as an academic than teaching into the void, teaching to a classroom that's completely silent, or teaching to a Zoom room in which no one's got their cameras on and you know half the people aren't even there. So I am genuinely particularly grateful to those of you who are brave enough to sit in the front row, and I was not one of them as a student, so I owe you double thanks for doing that, or keep your cameras on in Zoom meetings so I can at least see a face out there. Because it turns out, when you're teaching, you rely on seeing faces to give you a sense of whether the students are getting, whether the audience is understanding anything that you're saying. So thank you for allowing me, and more generally allowing all of us, to be part of your education, because it is genuinely an honor and a privilege to do that. I want to thank my fellow faculty too. I consider myself very lucky that I've worked in the Biostatistics division at USC, and the people who have run that division, such as Duncan and Jim and all the senior faculty who are mostly in that room now, I think, I owe great thanks to, because not only have they looked for people who are smart in the department, and that obviously counts, but they've also looked for people who are nice and on some level who are good people and who are collegial, which means we help each other. I have never had a bad experience in this department in which I felt like I've been fighting with my fellow faculty. When I read novels set in academia, that seems to be all they're about, but I've never had an experience like that in biostatistics, and I think that's great credit to everybody in the department, and in particular the people who are responsible for the hiring. So I thank them for that, and I advise you as students not just to focus on working with smart people or people who are achieving important things. It also matters how they conduct themselves in real life. So try to work for good people as well as smart people. And then just to wrap up, I've got 2 slides left. To return to life lesson 1, I feel like I've taken a lot of random jumps in my life, and I love random walks. It's one of my favorite bits of probability theory, so that's not a coincidence, I suppose. I remember when I was interviewing with Peter Donnelly as a potential PhD student, he asked me a question about random walks and whether or not they were recurrent, which means do they return to the origin infinitely often as a function of the number of dimensions that they're walking over. And it turns out, luckily I knew the answer, so he offered me a position. It turns out they're recurrent in 1 and 2 dimensions, but not in any higher number of dimensions. So it means that I encourage you to not be afraid to take jumps in random directions. But if you do that in a two-dimensional random walk, it does mean that you will return to the origin, back where you started, infinitely often, which is probably not a career path. So while I encourage you to take jumps and not be afraid to jump, you should at least give some thought into the direction that you do jump. You need to introduce some bias into this random walk to make sure that you're not returning to the origin infinitely often. And then I was trying to finally find sort of one summary overall life lesson. And as I was Googling around trying to find something I liked, I came across this guy who I'd actually never heard of, Howard Thurman, who was an American author and philosopher, born around 1889, if I remember rightly, and obviously dead now. And he is credited with this quote, which I really like. Don't ask what the world needs, ask what makes you come alive and go do it. Because what the world needs is people who have come alive. And I would add, that's what you'll really enjoy doing as well. I think that's fantastic advice. I didn't know of that quote until I was writing this talk, essentially. So somehow I sort of lucked into it. But that's, if I had to summarize my life lesson in one sentence or one phrase, one quote, that would be the quote. If you have the luxury of working in an area that you love and makes you come alive, and I've been very lucky to do that over my career, then you're a very lucky human being indeed. So look for that thing, of course, acknowledging that whatever it is has to also pay the bills. All right, that's what I've got. Apologies for using all the time, but I'll be happy to take any quick questions if there are any questions.
Any questions? Guys, online? I have a question for you, Paul.
Sure.
This may take longer. So in the world of machine learning and using cross-validation, I guess I've been thinking a lot about approximate Bayes calculations in that and how it can be used for some of these new large language models and other things where the likelihoods of those are intractable, but we can simulate from those. And a lot of times we do that with cross-validation, but maybe we should be doing it more formally. Have you thought at all about that?
No, I haven't, but it would work in the same way. So you'd be coming up with summary statistics or some other low dimensional summary of the data and using that as your sort of benchmark of success. I would just offer a caveat, which is that to this day, as far as I'm aware, there is still no theory, and I find it hard to imagine how anyone would ever write any theory, to quantify how close your approximate posterior is to the posterior distribution you actually care about. And if you can't quantify that difference, you're in a world of hurt, right? So what we end up doing in ABC papers is coming up with this set of summary statistics. We simulate large number of data sets and then run the ABC algorithm on those simulated data sets for which we know the generating parameter values and show that in some sense we do a decent job. And then we ask the reader to take a leap of faith and say, well, we did a decent job on all these simulated data sets for which we knew the answer. So now we're going to assume that we're doing a decent job on the real data as well. But of course, the real data didn't come from whatever that model was, right? All models are wrong, to quote George Box. Some are useful, and we hope this is a useful model. But you would really like to be able to quantify some sort of measure saying that if my low dimensional summary of the data is within a distance of delta of the observed data, then my posterior distribution is within a distance of epsilon of the actual true posterior distribution. I find it really hard to imagine that. I mean, there is a small literature in this field, but the papers I've seen only work in very specialized situations. So ABC is a method you use when you really can't do anything else in some sense. And for that reason, I'm somewhat dubious that it would be widely embraced by the AI community.
So one other one, Paul. So early on, how much was computational complexity a limiting factor, and did the advent of faster computing, in your view, contribute to the increase in the usage of ABC over time?
Yeah, no, absolutely that, right? So computational considerations have always been one of the chief limiting factors in everything I've done. And it never changes, right? As the computers get faster, you just do bigger analyses. And you're always, the limiting factor ultimately is your patience, right? I've always been willing to wait about 2 weeks maximum for some analysis to work. And if it takes any longer than that, so we used to wait 2 weeks for the MCMC things to run when I was first working with Simon. And if it took any longer than that, we would just give up and go home and find a different problem, right? So you, because computers get faster, you can do bigger data sets, but now you're now collecting bigger data sets, and the analyses are still taking 2 weeks, right? So in some sense, it helps, but in another sense, you always have exactly the same problem you had before. You can always collect more data. You'd always like to collect more data, and the analyses just get slower and slower as you do that.
Great. All right. Thank you, Paul.
All right. Thank you, everybody.